this webinar that the SEBBM is organizing with Patricia González as the coordinator of the Neurobiología Molecular group. Today, Patricia has invited Amanda Sierra. She will introduce her better, but for me, it's really exciting to have Amanda here because we met several years ago during COVID, and we worked together on that time. So it was really nice to have you here, Amanda.
Thank you.
So now Patricia is going to tell a bit more about Amanda, and welcome, Amanda.
Thank you.
So thank you so much, Amanda, for accept the invitation. So Amanda Sierra is a principal investigator and Iber Basque research professor at the Achucarro Basque Center for Neuroscience. And she is working on microglial cells and the interplay between phagocytosis and inflammation in the disease brain. She's doing a great job, and she has an amazing paper in the last year. So thank you so much for coming, and just waiting to hear you. Thank you so much, Amanda.
Thank you. Thank you very much, all of you, for the invitation. I actually don't belong here. Like I've been traditionally part of the Spanish Society for Neuroscience, but I recently joined the society. So it's really good to see that there are colleagues in both sides. So I'm very happy. I really think I belong here. So without further ado, so I have 1 hour in total with questions, right? Yeah, I'm usually on time, but if you guys see something wrong, just raise your hand, let me know, I'll be fine. So I'm gonna share my screen. It's here. Here. Okay, you can see my screen, right?
Yes, great.
Okay, perfect. So I will start. Okay, perfect. So my title is Brain Scavengers, Microglial Phagocytosis in Health and Disease. And let me put the pointer. So these, you have to get used to this because this is what we're gonna see throughout my presentation. This is microglia in the brain. They are beautifully ramified. And occasionally what we see them doing is eating up the apoptotic cells. So before we continue, I would like to, for those of you who are more interested in microglia, we co-organize with Lausanne University a European microglia webinar series that takes place every first Monday of the month. And we have an amazing lineup of speakers, past speakers, future speakers, and this is all deposited online in case you are interested. You can join us online or in different social media. So many of you are probably familiar with microglia. They are just the brain macrophages, more like macrophages of the brain parenchyma, because there are other cells of the macrophage lineage in the brain, in the meninges and so on. And among the many functions that you are probably familiar with, they interact with the neurons, they interact with the blood vessels, they interact with the astrocytes. So they are really pleiotropic in that sense. But so among all those functions, my lab focuses on understanding phagocytosis. So phagocytosis is really not like a state, but more like a process with different stages. So first, microglia, because they are motile, they are chemoattracted by different factors released by apoptotic cells, like nucleotides. That leads them to a specific recognition through a series of receptors. Then they form these 3D pouches that are called phagosomes that then fuse with lysosomes, and they are finally degraded. But what I'm mostly interested in is what happens next and how this process contributes to tissue homeostasis. As I was saying earlier, this is the way we're going to look at these cells. This is the mouse model that we usually use to visualize microglia. Most of the images are going to be in cyan. And here what you see is microglia standing this branch engulfing this apoptotic cell that you can see with this condensed DNA that we usually see with DAPI. And I like this picture because it really reminds me of a vulture. Like, I know this is a little bit stretched out, but you can imagine that these are like the wings and this is the neck, and this microglia is sitting up this apoptotic corpse. And I like this idea, even if vultures are not very pretty, because they are very useful. They really contribute to this, not tissue homeostasis, but the cleaning up of the ecosystems. And this is the idea that I'm interested in understanding, how they contribute to the cleaning up, to the maintenance of the brain ecosystem. But because it's sort of ugly, my students really wanted something a little bit pretty and more cheerful. So we designed Cookie, cookies for Cookie Monster, but also because microglia is super cute. And here you can see these microglia gambling all these apoptotic cells / cookies and eating up the apoptotic cell here in the stomach / lysosomal digestive system. So this is what we're interested in. So I'm going to do a brief introduction, and then I'm going to tell 2 unpublished stories from the lab that I hope you will like. So please do not share the data. Even if this is posted on YouTube, it's fine. So a few years ago, what we have been understanding in the last few years is the rules that govern microglial phagocytosis. And usually, I mean, it's actually simple to understand. Usually, you have very few apoptotic cells in physiological conditions, so you have few microglia engaged in phagocytosis. But when you increase the number of apoptotic cells, and we are going to see one example with a model of irradiation, what microglia does is they utilize different strategies, like recruit more phagocytic cells, even let's say 100%, increase the phagocytic capacity per cell, so each microglia can engulf more than one apoptotic cell, and in some chronic conditions, they proliferate. So by combining these 3 strategies, microglia can really cope with large amounts of apoptotic cells in health and in disease. Now, this is not working in some diseases where there's this uncoupling between apoptosis and phagocytosis, and we have done different studies in epilepsy and stroke. There is impaired recognition, impaired engulfment, and that leads to delayed cell clearance, inflammation, and further tissue damage. So one idea that I'm not going to be discussing today, but it's important to understand, is whether this process could actually be the target of future therapies, whether we can accelerate the clearance of the brain tissue by promoting phagocytosis. So now I'm going to show you precisely how we look at this process, and our favorite model is the hippocampus. The hippocampus, for those of you who are not familiar with the brain, is a region of the brain that is located here in the temporal bone, and it's really important for processes related to learning and memory. And one of the molecular cellular characteristics of the hippocampus that it's very striking is that the hippocampus contains adult stem cells. These are called the radial neuroprogenitors, and they are present throughout the lifetime of the animal and also in humans. And they give rise, they keep on proliferating throughout adulthood, and they give rise to granule neurons. What we discovered many, many years ago when I was a postdoc in the USA is that the majority of these cells undergo apoptosis naturally, and then they are engulfed by microglia without any need for activation or any kind of pathology. This occurs in physiological conditions at a very fast rate. So again, we're going to see here, this is with DAPI, all the neurons. This is the radial neuroprogenitors. This is one daughter cell that is much smaller than these ones, and it's because this one is apoptotic. So this is a newborn cell that is apoptotic and is engulfed by microglia. So you can really see that life and death intermingle in this neurogenic niche. And if you look at this in 3D, you can really appreciate that microglia do not have this conventional amoeboid morphology. This efficient phagocytosis is executed by terminal branches of microglia forming these 3D phagocytic pouches. So from here, a few years ago, we started wondering, so what? What's next? And in order to understand how could these phagocytic microglia contribute to the neurogenic niche or to homeostasis, we started by running some very old gene arrays in culture microglia. So we fed microglia with apoptotic cells. You can see here that they really engulf them, they become phagocytic, and we could find many genes that were upregulated in the phagocytic microglia. Interestingly, some of these genes were related to neurogenesis and neuroprojection development. So it's not a unique gene in phagocytic microglia that is related to neurogenesis. We have peptides, hormones. This is 3 hours of phagocytosis and 24 hours of phagocytosis compared to controls. We have trophic factors, we have matrix enzymes, we have cell surface ligands, and many cytokines. So it's not that just one single molecule could act from the phagocytic microglia to the neurogenic niche. It's more like a bunch of substances, most of which are released. So this is why to really demonstrate that the phagocytic microglia contributed to the niche, I'm going to summarize really briefly because this is published many years ago, but we focus on the secretome. We focus on genes whose product would be secreted and could act back like a feedback mechanism on the neurogenic niche. So I'm going to summarize 3 types of experiments that we did. First, we collected the secretome, or let's say the conditioned media from phagocytic microglia, and we use it to culture neurospheres, neural stem cells. So you could see there's a dramatic change in the aspect and the markers expressed by the stem cells when they are cultured with the phagocytic microglia conditioned medium. And through many types of experiments, we demonstrated that it turns the stem cells into the astrocytic lineage. When we put this conditioned media in mice, we could also show that it decreases the number of newborn cells that are labeled here with double coating. So it seemed that this was like a negative signal coming from the phagocytic microglia to the neurogenic niche. But things get a little bit more complicated because we use different constitutive and inducible models to specifically block phagocytosis by acting on the P2Y12 and the MERTK signaling pathways. So here, what we could see was very striking. When we looked at the acute model, so if you just block phagocytosis, what you had is an increase in neurogenesis, which was really consistent with the block of neurogenesis that we could see in these other 2 experiments. But if you waited longer, what happened is that the initial increase in neurogenesis actually was not sustained over time. It really went down even below basal levels. So we found this biphasic regulation of neurogenesis when we blocked phagocytosis. So somehow this negative feedback loop was necessary for the long-term maintenance of neurogenesis. So this is giving rise to this idea that phagocytosis has some sort of homeostatic control of adult neurogenesis in the long term. Now, we followed up, we started these experiments a few years ago, and this is going to be one of the first unpublished stories that I'm going to tell you about, but wanted to go even further. And in order to do that, we acknowledged that there were some limitations in the in vitro study, so we wanted to study the consequences of phagocytosis in vivo. And to do that, we started a collaboration with Inigo Casafont in Cantabria, and we were using his irradiator to irradiate the niche. This is again the neurogenic niche. This is the hippocampus, and right here, and this is the area in the subventricular zone, subgranular. This is where the newborn neurons should be produced. But when we irradiate the mice, this is LCA for low cranial irradiation. What you can see is there are many apoptotic cells labeled with activated caspase-3 lining here, right in the neurogenic niche. I hope you can also appreciate it here that this is super specific. So it's low cranial irradiation. We use a very low dose. It's only 2 grays, as little as we could. It has some side effects we can discuss later, but basically what you can see is there's an increase in apoptosis at 6 hours, and everything has disappeared by 24 hours. So there is full clearance. And why is there full clearance? Because microglia are engaged in clearing up all the apoptotic cells. Actually, if we calculate the phagocytosis capacity, which is the percentage of microglia with more than 1 pouch, what you can see is that at 6 hours, the majority of microglia are engaged in phagocytosis with up to 5 pouches, and then this goes back to normal, like the majority of microglia being non-phagocytic later on. Here's an example. This is very messy, but this is how the model actually is. It's super messy because there are so many apoptotic cells that it's really difficult to analyze. For instance, this cell up here, which is labeled in cyan in our mouse model and also expresses a microglial nuclei marker, Q.1, is that, it's stretching this process here, engulfing this cell, really distal cell here. And this other mess here is shown here. You can see a microglial nuclei eating up all these apoptotic cells. So in the end, what happens is that the phagocytic capacity increase compensates the increase in apoptosis, and this is why we have full clearance. So they really can eat a lot. Now, it's important that you realize that this is specific of the dentate gyrus. It does not happen in other regions of the hippocampus like CA. Like that would be up here. So if you look at CA, we have a little bit of apoptosis. It's like 20 times less than in the dentate gyrus. And actually, there is no increase in phagocytosis in CA at all. So this is important because now we are going to be able to identify effects that are due to irradiation, which would happen both in the dentate gyrus and CA, and effects that are specific to phagocytosis, which occur only in the dentate gyrus. So using this model, now that we have analyzed it, we performed single-cell RNA sequencing with the help of Marisol Aymeric, and all the analysis was done by José López Atalaya in Alicante. And this is what we could see. So there's many clusters as usual, and it took us a little bit to identify which clusters were relevant, but basically there is two. There is this light blue that is called lysosomal microglia, which really increases over 24 hours, and the proliferative microglia that we will see later on, which increases at 7 days. So this lysosomal is not that different from other clusters, but the major difference is the expression of LGAL-S3, which is a gene that our colleague José Luis Venner is an expert on. This you can really see here increase at 24 hours, reduce sort of to normal levels by 24 days. So what is galectin-3? Is super pleiotropic molecule related to many diseases, many cell functions. And we find it's upregulated specifically in the post-phagocytic microglia, so 24 hours and even up to 7 days, again, specifically in the dentate gyrus after irradiation, not in CA, so that really points towards its relationship to phagocytosis. So it's one of these post-phagocytosis adaptations, and where we wanted to know what could be galectin-3 relevant for, José Luis was kind enough to share some of his galectin-3 knockout mice, and what we have found super recently is that these mice have impaired neurogenesis. So here you can see wild type and knockout 24 hours, no big differences, but at 7 days, the neurogenic niche is recovering from the irradiation only in the wild types, not in the knockouts. And specifically, if you look at the proliferation of the radial glia, which is shown here, so here you have the two proliferative, this is in anaphase when they are separating, you can see the chromosomes, and you can see a little bit, it's difficult to see, I know, but this is how difficult this is to analyze. You can see the apical process of the radial neuroprogenitor labeled with nestin and GFAP, without changing in the total number of radial cells. And because there is more radial neuroprogenitor proliferation, there is also more production of young neuroblasts. Again, here with double cortin, we can seal the neuroblast, and I think it's evident that there's a lot less in the knockout mice than in the wild types. And you can see again this recovery of neuroblasts, these mature ones, which are the first ones to be produced, and overall in all of them. So the conclusion from this part is that at least galectin-3, so phagocytosis triggers some adaptations, including the overexpression of galectin-3, which is then responsible of this regeneration of the neurogenic niche by the post-phagocytic microglia. Then we looked at the other cluster that appear upregulated, mostly at 7 days, which is the proliferative cluster, both cells in S phase and G2M. And we validated this in tissue using Ki-67. Here you can see this is an intact neurogenic niche. At 6 hours, it's all gone. Basically, you only have apoptotic cells, no proliferation, which is slowly coming back, right? And some of these cells, like this one here at 30 days, is proliferative microglia. So proliferation comes back. Microglial proliferation appears delayed compared to the neurogenic niche. And again, this is specific of the dentate gyrus and does not occur in CA, so it's really specific of post-phagocytic microglia. However, what is this proliferation doing? We actually don't know exactly, because you don't have increased numbers over time. So the total number of microglia is sort of constant. We have this decrease at 24 hours, but there's no increase over time. So we started wondering, is this real proliferation, or is just like some sort of aberrant expression of Ki-67? So to further confirm that there was indeed a proliferation, we injected the animals with BRDU, which is an analog of thymidine that it's incorporated in the DNA of dividing cells, and then we sacrificed the animals at 2 different time points. So we could see proliferative microglia in the 2 time points. This is total number of BRDU cells, which is sort of constant between these 2 time points, but the number of microglia, of newborn microglia, decreased. So this suggests that there is some sort of cycle of cell death and proliferation, so that maybe there's initial cell death or stress accumulated over time. This triggers proliferation, but this proliferation it's aberrant. It's not driven to increase the number of cells. We speculate that this is related to some sort of renewal of organelles. And why we think that? Because we have found out that phagocytosis is actually very stressful. So here you have an example of a phagocytic microglia that it's dying. So here's microglia. Here with caspase-3, you can see that not only the apoptotic nuclei clearly included in the phagocytic pouches are labeled with caspase-3 and are pygnotic, but also the microglial nuclei itself. This is very difficult to quantify in vivo, so we turn again to our in vitro system where you can really see that apoptosis is triggered when you treat microglia with apoptotic cells. So once they become phagocytic, some of them will enter apoptosis. Why? Well, they have increased radical oxygen species, so they have oxidative stress. They also have a lot of changes in many catabolic pathways. For instance, the glycolysis is actually downregulated. Mitochondrial respiration is also going down, but they also have other pathways that are increased. Like, for instance, the production of spermidine, which is antioxidant, it goes up. Whereas spermine, which is the product, goes down. So there's really a huge metabolic rewiring during the post-phagocytosis stages. And we have even found out that there is a lot of remodeling in the mitochondria. So, I mean, I'm just showing you some electron microscopy images, but of course, this is all quantified. Usually what we see is that naive microglia have many mitochondrial objects, so because they are super branched, but the post-phagocytic microglia is not. They have fewer mitochondria and they are more simple. They are less ramified. So this triggered the idea of, okay, all these changes, are they going to compromise microglial function? And for this reason, we decided to re-challenge microglia with a second apoptotic challenge. So in physiological, in our model, we did 2 doses of low cranial irradiation separated by 7 days. So we sacrificed the mice after the first one or the second one, 6 hours to see all the production of apoptotic cells, and 24 hours later to hopefully see the clearance. So what you can see here, sorry, this is a little moved. Microglia engulf the same percentage of apoptotic cells, suggesting that there really is no change in the phagocytosis efficiency, even when they are suffering all this challenge and all this metabolic rewiring, metabolic stress, and so on. However, there is an issue with this experiment, if you see here. During the second challenge, the induction of apoptosis is much lower than during the first one. And this is because the neurogenic niche is still compromised. It has not fully recovered. And because this is not really doable in these physiological conditions, we started a collaboration with Dolores Hamatsobian from Mount Sinai, where we use the same kind of experimental design, 2 challenges, but now we're using a model of glioblastoma. So now we use a higher dose of irradiation, which is what is conventionally used in these models. You can really see it's a very complicated model. Please don't ask me too much about it. But basically, they have this neurofilament type of neuroblastoma, huge damage, I mean, huge tumors in the brain. You have all these tumor-associated microglial macrophages. You have some cases of phagocytosis, not as many as in vivo, but again, sort of the apoptotic challenge was the same size, and again, microglia had a similar phagocytosis efficiency. So overall, this suggests that these phagocytosis-induced changes are adaptive and sustain the function. So in summary, this is work by a fantastic PhD student in the lab, Mark Marquez. What we have shown is that phagocytosis does not end with the digestion. What you actually have is a lot of changes from P2Y12 clustering that I didn't show, transcriptional changes, metabolic alterations, mitochondrial remodeling, cell death and proliferation, which ultimately lead to sustain their function in the long term and promote these regenerative properties of microglial phagocytosis. So I'm going to stop a little bit. This was the first study that we did. We're going into publication into submitting the paper soon. And now I'm going to tell you the other study. It may be a little bit longer, so I may have to skip a couple of the last slides, so to have time for questions. So now I'm going to go to the other way. So what I've shown you so far is what happens after phagocytosis. Now, what we started asking is what happens before phagocytosis, actually what happens during development, when microglia really enter the brain. So this is what I've shown you so far. And we had this observation in a supplementary figure long ago, showing that from postnatal day 7 to postnatal day 14, all the way into adulthood, there seemed to be an increase in the percentage of apoptotic cells that were engulfed by microglia. So we started wondering, are they really efficient phagocytes when they enter the brain parenchyma during development? So you probably know that microglia come from the yolk sac, and they see the brain. We know a little bit of the transcription factors that drive this process. They start colonizing. And then what we really don't know much about is what happens once they enter. Because we know that in the early embryos, what we have is very few microglia. They are sort of amoeboid. They are really dispersed. And we end up with a brain in which microglia is perfectly tessellated, perfectly ramified, and we know they are very efficient phagocytes. So we wanted to understand this process. So what we have started to understand is by trying to use a mathematical model to understand how the colonization is occurring. So I'm going to show you a bunch of numbers. Don't look at them too much. Basically, most of the studies that I'm going to show you have been done both in the hippocampus and cerebellum, but I'm just going to show the hippocampus data. Basically, what happens during postnatal development is that the hippocampus is growing. At the same time, the number of microglia is going up until a plateau around P21. But the density of microglia decreases a little bit until P60 because the numbers are constant, but the hippocampus keeps on growing. This is a little bit difficult to understand, to be honest. And at the same time, if you look at microglia proliferation with Ka67, what you see is a really early drop in proliferation around P4. So how come they reach the total number by P14, but they drop proliferation at P4, the density increases that then goes out, down. This was so complicated to understand, and this is why we decided to start a collaboration with Jose Carrillo and his fantastic team in Oxford and Decam. So we used different models to try and see how we could understand microglial colonizations and how all these numbers came up together. So the first model only took into account proliferation and apoptosis and the volume, increasing volume of the brain, and didn't work. Then we made another model that was more complicated, which would have both proliferation, apoptosis, but also migration from other sources. And again, didn't work. So finally, we opted for a model. It seems a little complicated, but just follow me, you will understand really quick. Basically, we allowed microglia to distribute into two mathematical populations, population A and population B, that were completely independent. Well, A could transform into B and B into A, but basically each one had its own rates of proliferation, its own rates of apoptosis, and its own rate of spreading. Now, this model was really good. Now, these are four different mathematical criteria to assess which one of these three models was better. And the higher the number, the better the fitness. You can see that this two population model was much better than any of the other two fit in our population. So what does it mean? So you can see here the actual fitness of the model. It's really good. In blue, it's the actual data, and in the shaded area in green is the simulations. And it's really, really good fitting both the region of the hippocampus, the density of microglia, and the percentage of proliferative microglia. So with this, sorry, this is a mistake because this is not percentage. So what are these two populations, right? To do so, to understand this question, what we use is Monte Carlo simulations to estimate the values of these parameters, the proliferation and differentiation and so on. So this looks a little bit complicated, but if you can follow me, you will understand that proliferation of A cells is really high, whereas proliferation of B cells is really low. So A cells are proliferative, B cells are not. And proliferation is largely driven by intrinsic factors because this other parameter is much lower. In contrast, the differentiation is mostly extrinsic, and again, is higher for A turning into B than for B turning into A. So now our summary or model is much more simple. Basically, we have a population of A cells that are highly proliferative and turn into B cells that are not proliferative and do not differentiate back. So I'm quite astonished, actually, that this modeling discovered a very simple biological principle that is called terminal differentiation of a progenitor. Basically, again, we call them P cells for proliferative, and the B cells are Q for mitotically quiescent. And if we plot them over time, you can see that they don't really coexist. one really transforms into the other. So early on you have the red cells, and later on they transform into the blue cells, the quiescent cells. So now, so the model is really a discovery of a critical event in the lifetime of microglia, which is the synchronized turn-off of proliferation into mitotically quiescent cells. And now we can start, which occurs really early on between the postnatal day 3 and 4 in the hippocampus. So now we can start asking questions about what controls this switch and what is this switch related to. So one of the model has other predictions. One of them, I'm gonna go a little quick over this, but we can discuss later. One of them is whether there's to which extent dispersion is relevant. There's a little bit in the hippocampus. The dispersion goes from the CA into the dentate gyrus, whereas in the cerebellum is the other way around, from the white matter to the external molecular layer. We also know that there is very little role of apoptosis because the rates are very low. The model has a very interesting prediction, which is that the microglia doubling time will increase over time. So to validate this, we calculated the cell cycle. To calculate the cell cycle, we injected 2 analogs of thymidine, EDU, and BRDU at different sequences. So for instance, you can see here, this is a microglia that was labeled with EDU and BRDU, so it re-entered cell cycle. Whereas here you have 2 microglia that were labeled with EDU, but did not re-enter cell cycle. Now, when you analyze this over time and quantify this, when there is a peak of colocalization, the interpretation is that one cell cycle has been completed because the cells have re-entered PS phase. So basically with this type of analysis, we can assess that at P2, the cell cycle is really, really fast, around 12 hours, whereas by P7, it's already lengthened by 24 hours. And this actually validates this doubling time prediction of the model. We have also observed other modulations of microglia during this period, particularly in the metabolic profile and the lipidomic profile. Metabolic profile is kind of ugly, so maybe you just look at here. Basically, you can see P2, P7, and P14. They separate really, really well. The clusters really work really well. So if you wonder, basically, because there is all these ugly names, basically what you have here is phosphatidylcholines and serines at P2 and P7, and which are more like structural lipids. And at P14, what you have mostly is triglycerides and diglycerides, which are mostly related to energy accumulation and consumption. So they're really, all this switch in this very, very short time window from the cells being proliferative until the cells are post-mitotic, it really is accompanied by a huge remodeling of their metabolic profile. Now, what else? So what is the consequence? What happens when cells stop proliferating? Well, only when they stop proliferating is when they become phagocytic. So if you, now we analyzed a number of apoptotic cells over time. There is, in the hippocampus, there is an early peak at P2. This is the developmental peak, and another peak at P21, which is the one related to the establishment of the adult neurogenic cascade. But you can see how they start eating more and more and more over time. You could think, okay, maybe Amanda, they are not eating at P2 because they have too many cells to eat. But this is not true. If you calculate the size of the challenge, how many apoptotic cells per microglia, at P2 they don't have even 1 apoptotic cell per microglia. So that's not a lot. If you compare with the data that I just showed you from the irradiation model, in the adults, microglia can easily eat up to 4 cells on average. And they are fine. Well, some of them die and so on and so forth, but they can really cope with huge number of apoptotic cells. But at P2, they don't. So it seems that they are really not born efficient phagocytes. To further confirm this is a complicated experiment, so I'm gonna try to make it easier for you. We challenged microglia with more apoptotic cells using ethanol, both at P2, well, at different time points, using low dose and high dose. So for instance, if you compare here, apoptosis at P7 at P14, you can see that the low dose of ethanol induces a similar challenge. Again, similar ratio of apoptotic cells per microglia. Microglia react similarly, but not enough. At P7, they just don't eat enough. And many apoptotic cells remain unphagocytosed, whereas by P14, they can cope with everything. Same things happen if you use a larger dose at different time points. Now it's P9 and P14. Again, you induce a challenge, similar size. they both sort of react, but the reaction of the P9 is not sufficient, and a large number of apoptotic cells remain unphagocytosed. So definitely they are not born efficient phagocytes. And why is that? Well, we don't know. There's probably several reasons, but they are not born efficient phagocytes. One is the morphology. They are really much more complex over time. You can see they have more branches, longer branches, and they use the branches to find the apoptotic cells, right? So it makes sense. We also know from that they survey more area. If you do two-photon imaging, you can really see that by P14, they can really read their parenchyma, their surrounding parenchyma much better. But also many genes involved in sensing, sorry, there's something missing here. Many genes involved in sensing increase their expression over time. This was already known, so this is not really surprising. And together, all this data really explains why microglia are not born efficient phagocytes. But over time, what you can see in this cartoon summarizing the results, basically we have two phases. We have one phase where microglia proliferate, and then everything else happens. The volume of the hippocampus increases, they increase the morphological complexity, they increase their number, they become efficient phagocytes. So it really seems that there's two programs, or at least two stages in their development: proliferation and functional efficiency. Actually, if we have correlated all the data over time, and as you can see here, there is a very strong negative correlation between phagocytosis and proliferation. So we started wondering, are these two exclusive? Can the cell perform the two events, the two processes at the same time? So we've done several experiments to try and understand this. This is, I think, the most relevant one that I'm going to show you. So one of the major pathways controlling proliferation is the CSF1 receptor, colony stimulating factor receptor. And it has two ligands, interleukin-34 and CSF1. So we can block this pathway using this drug, GW2580. Okay, so this is the experiment. Now you can see that if you use this drug from P2 to P5, you reduce proliferation. The effect is not so evident at P7, but in both cases, you reduce the number of microglia. Maybe you can forget about this group, just skip it. If necessary, we can go later on. This effect does not affect phagocytosis, but it does affect apoptosis, but it does affect phagocytosis, suggesting that if you block proliferation, you end up with less phagocytosis, suggesting really that these two processes are linked. And the cell cannot really do one and the other at the same time. So actually, this seems to be a conserved decision in other paradigms. For instance, with Alberto Pascual in Seville, they have used a well-known paradigm of microglial depletion using Plexicon. So you can really deplete microglia and they repopulate the brain. And again, you can see that early on they proliferate, and only when proliferation is complete is when they become efficient phagocytes. Again, showing this really negative correlation between phagocytosis and proliferation. We have also tried to analyze this in other tissues, and this is a collaboration with David Menasa from Oxford University. This is human brain from the 17th post-conceptional week. I have to say we have very few cases here, so because this is very challenging to do. In this tissue, what you can observe here is this is the hippocampus, and here what you have is the ganglionic eminence. And here in pink, you can see IBA1. So there's different things that we can discuss. There's some hotspots of microglia. Maybe they are proliferating, I'm not sure. But basically, we decided to look in all these regions, and we combined all the regions, basically, at proliferation and phagocytosis. And here are some examples. So this is not confocal imaging, right? So you cannot, because there's a lot of ethical difficulties with obtaining and analyzing this tissue. So the images were actually taken by David and they were sent to us. So we just analyzed these bright field images. And so what you can do is to do color deconvolution. So in color deconvolution, you can separate the different channels and have an idea of colocalization. But I have to say it's not definitely as good as it is with confocal microscopy. So there's a limitation in the study for sure. But basically, this would be two microglia that just proliferated with Ki67. You can see this really dark nuclei. Here you see an example, for instance, in the ganglionic eminence of 17 post-conceptional week. Here's a dim apoptotic cell. here's a microglia, no phagocytosis. This is probably one of the more clear examples of microglial phagocytosis extending the branch, engulfing this apoptotic cell. And there are some cases that we have labeled as apoptotic microglia because there's like perfect colocalization, but we have not quantified this. It's very difficult to observe. Now, if you pull together all the regions and the 3 ages that we have assessed, 16, 17, and 19 post-conceptional weeks, what you can really see again is this negative correlation between proliferation and phagocytosis. So in later stages, microglia are more phagocytic and less proliferative, also in the human brain. So finally, I'm gonna go a little bit quickly on this. Maybe I should skip it. I'm just gonna say a couple of sentences, I think. But basically, we started looking at how is this controlled. And we started, just skip this, we started looking at epigenetic marks because we could really see that there is differential enrichment in methylation in different genes that are associated to this epigenetic mark, histone 3 methylase, which is specific of active promoters. One of them is super interesting, we can discuss later on, is ApoE. What is ApoE? Really going down in microglia around the time when they switch from proliferation to phagocytosis, ApoE being a well-known Alzheimer's risk gene that maybe you want to discuss later on. But basically, we wanted to really show whether this methylation could control microglial development. So we found a very interesting pathway, which is called Icaros. So Icaros is an inhibitor of a methylase that acts on this mark. And why is Icaros interesting? Because it's mainly expressed by microglia, and the group of Albert Giral had already shown that it had effects on adult microglia. So we started a collaboration with him. And again, what we could see is that Icaros knockout mice had reduced numbers of microglia, impaired proliferation, and really reduced phagocytosis. So again, suggesting this link between proliferation and apoptosis. So just to summarize, this was mostly done by, again, a super talented PhD student, Marta Pereira Iglesias. Basically, we have shown these developmental milestones of microglia from proliferative cells to efficient phagocytes. This process is controlled by Icaros, and it's accompanied by this switch, a synchronized switch, in which what we have is accompanied by changes in cell cycle dynamics, in the metabolic remodeling, in the epigenetic landscape. And ultimately, what we have shown is that if you block phagocyte proliferation and colonization, you will not have efficient phagocytosis. So overall, this suggests this really dilemma between proliferation and phagocytosis that may be relevant for neurodegenerative diseases. So I'm just gonna wrap it up with thanking everybody. We had a lot of collaborators. I'm super grateful for all of them, both in the paper with the adaptations and the developmental paper. Everybody in the lab did a lot of work, Joel, Marco, Xavi, and Lucia. And thank you very much, the founders and everybody for your attention. Thank you very much.
Thank you so much.
Okay, so
Thank
we
you so
have time
much,
for questions. Sorry, I had to shorten it up a little bit.
No, amazing work. So I think we have a question in the channel, so I can do. Or Antonio, Miranda, do you want to
yes,
do?
No, you ask the questions, Patricia.
Okay.
That are written. And people should ask the questions on the chat.
Okay. Amanda, does microglia only phagocytate apoptotic cells, or does it with other dying cells, like, for instance, ferroptosis undergoing cells?
Yeah, that's absolutely right. But we don't know. So presumably, yes, but it's difficult to assess in vivo. So I'm sure if you feed them in vitro, they will probably eat them up. I don't think there's any comparison of how efficient they are. So the interesting thing about apoptosis is that it's very developmentally and evolutionarily conserved, right? So really microglia have a lot of mechanisms to find and recognize. They are really overlaying mechanisms devoted to recognize and engulf apoptotic cells, tons of receptors. For the recognition of ferroptotic cells, necrotic cells, is not so clear. So it's possible that some mechanisms are shared and some others may not be. My problem with this is that, not that I have a problem, but the problem of assessing or answering your question is that in vitro, yes, you can really assess necroptosis. In vivo, if I understand correctly, it can be a little bit tricky. So, but if you have an idea, just email me and we can try and help.
Thank you. I think that we have further question just with the hand up, but I cannot see the name. Just
There's
Can-
the
yeah, because Juan, go ahead.
Okay. Hi, Amanda. Congratulations. Very nice
Thank you.
talk. Hola. You show in the first step a change in phagocytic capacity and how they adapt after phagocyting the apoptotic bodies.
Mm.
The second, you show how microglia can evolve, can maturate through development. My question is, how specific are both changes of microglia, and that happen in the same way in peripheral macrophages? You can see
Oh, super interesting question. Yes.
Also to check as a biomarker for the disease, like the others one, I mean.
Yeah, like totally. It's just I don't have the expertise, but I really always wondered to which extent are microglia comparable to other phagocytes. So there's some experiments about some metabolic adaptations, particularly in the mitochondria, by Cody Ravichandran, is the super expert in phagocytosis in Virginia. And he was the first one who showed a few years ago that phagocytosis triggers metabolic adaptations that are necessary for the continuous uptake of. So I think there's probably going to be some shared principles. The developmental part, I'm really curious about, because no one really talks about phagocytosis efficiency in macrophages. So if there's any immunologist out there who wants to check this, I will be very happy. I think that there may be, I think that probably the main principles are shared because they are very similar. But I think there will be tissue-specific and target-specific adaptations. So I think eating an apoptotic cell is probably different from eating, I don't know, myelin deposit, let's say, or amyloid beta. Now, whether it's different eating an apoptotic neuron or an apoptotic hepatocyte, I don't know. It may depend on the, the lipid content or... So I think there will be some specific adaptations in the tissue, but probably the principles are the same. But really good question.
Okay,
Well,
thank you. Bye.
So we have further question in the chat.
yes. Well,
Patricia, I think
yes,
always.
yeah.
Oh.
I have two questions.
Yep.
One would be that yes, we were saying that there's a metabolic adaptation. This happens when cells are stressed, and many cells, parasites are what, and that there are catabolic reactions that are going on. So there should be more ATP. This is like a survival, or have you measured ATP and then?
You mean within the cell or in the?
Yes, within the cells or in
No,
that region.
we haven't, but it's a good point.
And does this affect the neurogenesis? I mean, the metabolic changes in microglia, does this affect directly to neurogenesis?
So we have not tested directly that hypothesis because, but we are testing it now, not in the context of neurogenesis, but in the context of glioblastoma. So in glioblastoma, you have a very sort of equivalent situation than the neurogenic niche. What you are doing with the treatment is you are inducing death with the chemotherapy or radiotherapy. You are killing glioblastoma cells. And then what's going to happen is that you are going to have all the tumor-associated macrophages eat all those apoptotic cells. So there's going to be metabolic adaptations. We are trying to identify which specific pathways are and which specific metabolites are released.
Yes, exactly. That's what we see in liver. So at the end, what you have is that what the glia or the microglia is releasing, that's where I wanted to go. I mean, you have that metabolic rewiring with all the secretion of metabokines, metabolites, or others that will affect other cells. So this is really important.
Yeah, exactly. So this is exactly what we're doing right now, but not in physiological conditions. We're moving to glioblastoma because it could have a, like maybe we could identify, because it would be terrible. Imagine if the tumor macrophages are eating up the apoptotic cells and then releasing nutrients, metabolites that are somehow fueling up the rest of the tumor cells. That would be terrible, right?
No, that would be, and it makes sense.
So we think there's probably a mix of metabolites. Some of them, like we have identified one in the mouse microglia. So we're doing this in human microglia with human glioblastoma cells. But in the mouse microglia, we have identified one pathway, which is the spermidine, which has already been shown to modulate tumor growth. So we do have some evidence that spermidine from the phagocytic microglia can modulate tumor growth, but we want to be more specific and really.
At the end, that's the methionine cycle also. And then you have the generation of glutathione in the same, because
yes, exactly. So what we need to find out is exactly what is released. What we have analyzed is what is inside the cell, but we have to figure out which ones are released.
And then the other question is that it called my attention really that during development, you have the first part of proliferation, and then you have like more like steady part. And then the accumulation of glycerolipids, however, you have it at the end. Because when you have proliferating cells, I don't know in the brain, but in other parts of the body, usually you have all the accumulation of lipid droplets, which are full of glycerolipids, triglycerides, and usually the reason is because you are using them. They will disappear because you are using them for the generation of new cell membranes, and all those glycerolipids will be used for the synthesis of phospholipids. So I wonder if during that proliferative phase, because what I see is that you have there the phospholipids or glycerophospholipids, but then at the end, the accumulation of those neutral lipids, maybe you have more lipid droplets at the end. You should have the activation of other metabolic pathways such as fatty acid oxidation or something so that you don't accumulate those glycerolipids. So do you know something about that? If
no, that's a great question. So we don't know. So two things. One, I would really love to look at lipid droplets. We're gonna do some electron microscopy in this tissue with this time point. So maybe we could see something. I don't know. So our initial idea was that maybe we don't know if these metabolic changes are adaptations or are drivers of the proliferation.
Well, probably both.
Probably both. So, but this is we honestly don't know how to manipulate it. So, if you have an idea, I would be happy to share the data with you. Maybe you can help us with this and see if there's anything that can be done.
I know how to work in other kind of cells. I've never worked with microglia or something. So if I can help, of course. And then this is a curiosity because I see, of course, there is ApoE, but ApoE is into lipoproteins. So this could be the way that there is the uptake of lipoproteins so that you have that accumulation of glycerolipids. But how come lipoproteins be there? Is there a lipoprotein-like particle that, because they don't get into, lipoproteins are synthesized in by the liver. So do they get into how do they get into the brain? Or what kind of cells are doing the synthesis of these lipoprotein-like particles? Because otherwise, how does the ApoE get there?
Yeah, that's a great question. I really have no answer. So there is so much talk, and I don't remember. So it's great to talk to you because you're the expert here. I don't remember any single paper discussing this, but ApoE is this risk factor for Alzheimer's. So everybody is focused on ApoE isoforms. We are doing some work with ApoE isoforms, but there really is no discussion of how or what is the actual mechanism through which the ApoE, and whether it's related to lipoproteins or something else, is actually affecting microglia.
Like a bunch of years ago, like in 28 or something like that, I remember when I was in Canada, there was this group that I don't remember the name, that they were working, because you were talking and I was thinking about that, and I remember that they were saying some kind of lipoprotein-like particle, because that could be the one that will be internalizing, to have an increase of that kind of lipoprotein, and that what could be the one that will be driving all the lipid supply, so that maybe, I don't know if that would be important or not, as you mentioned, a trigger or a consequence or whatever, but this is like
Yeah.
some idea I had of it.
Yeah, no, that's a great point. Do you mind if I write you an email?
No, write me, of course.
Maybe you can help me with it.
No, I think it's really interesting what you are seeing, because there are lots of pathways that we
there
have
are a lot
studied
of
before in
pathways.
other parts, but super interesting.
Okay,
Yeah.
thank you very much. You will get my email.
Yeah,
Thank you.
so we have some questions from the chat. The first, I mean, the second one is Miguel Ángel Burguillos. I don't know if you want to switch on the camera, but the question is regarding efficiency in the pH index. Have you measured somehow in the efficiency in digestions?
Digestion, yeah, that's a great question, and we have. So it is difficult. So in vitro, yes, you give a pulse of the apoptotic cells times 0, you remove them, you wash them out, and you wait, and then you can really see digestion. That's easy. In vivo is much more complicated, because in vivo what you have is like a single shot, right? Like a snapshot of whatever they have ingested, and that's it. But we made use of a trick that we used in the past, and it's not directly the digestion time, but the clearance time. So not really looking at what's inside microglia, but at the total number of cells that are present and how they disappear over time. So because the majority of the cells that undergo apoptosis are newborn cells, you can label them with EDU or BRDU. And then that's a spike of apoptotic cells. And then similar to this track that you can do in vitro. And then you can quantify how many you have a few hours later. So with this, we have estimated that in the adult, we had long estimations from long ago. It's like 90 minutes. In development, in P2 is like 3 hours. So it's not that there's no phagocytosis, just this is lower. Yeah. So I did not answer how the efficiency in digestion, but the clearance, yes. Clearance is delayed, yes. I didn't show the data because the analysis is so complicated that it would take me forever, but yes.
Okay, so the other question is coming from Robert Josep Subirana. Great talk. Did you see if microglia were affected by overphagocytosis?
So this is a great question. We have not directly assessed it, but I think it's what the data is hinting at. So I think the more cells you eat, I'm saying we did not directly test it, but I think everything we see in vivo and in vitro is because we are really overfeeding the cells. So I think there's probably some sort of threshold. So the more cells you eat, the more stress, the more oxidative stress, and the more probably metabolic adaptations. So yeah, we didn't assess it directly, but I would think it's like a dose-related effect.
Okay, thank you. Lola Martín de Saavedra, again, if you want to switch on your camera, but the question is, thanks for a great talk. I was wondering if you will expect to see sex difference in how microglia evolve or transform from colonizing to becoming phagocytic.
So that's a great question, but and there's a lot of literature suggesting some differences between male and female microglia. But in my perspective, I did my PhD on neurosteroids, so I'm very, very familiar with all these male versus female differences. And honestly, I think it's modulatory. It's not that major. Even with those papers that claim differences, it's more like a modulatory effect. It's not super robust. So no, we have not seen, we always track the sex of our animals, and we have not seen major differences in males versus females. I mean, if you look for them and you have large groups, maybe you will be able to detect them, but not very strong differences, no.
Okay, and I think that the last one probably, thank you, very interesting talk. Hygul secretome chain where when there are other phagocytic targets except apoptotic cells, also when the phagocytosis of them falls.
Yeah, the second question, can you rewrite it again? I'm not sure what
the second
no,
question
the writing. I need the person, Parisa, to rewrite it because I'm not sure what she means.
I don't think that is Parisa or we don't have the name. Patricia Aspichueta?
If someone can, so the first part is yes, I think.
Parisa Dastestani.
Yeah, if
Okay,
if you're there and you want to rewrite your second question, please do it. The first part is, yes, I think the secretom will change, because I think we have not tested it, but it's a question that I get regularly, and I'm pretty sure the metabolic adaptations depend on the target. Completely. It's not the same phagocytosis in a microbe, on a apoptotic cell, alpha-synuclein, beta amyloid, synaptic debris is going to be completely different, because in the end it's a bunch of metabolites. You have lipids, you have DNA, you have proteins that you have to digest, and somehow you have to do something with them, right? You cannot
no.
Yeah, so when the phagocytosis of them fails, I'm not sure what you mean. Yeah, they accumulate, that they just take longer. So, but if your question is what happens if an apoptotic cell is not phagocytosed, is that it transforms into a secondary necrotic cell and starts releasing intracellular contents that then are toxic. So yeah, you do need efficient phagocytosis, if that was your question. Not sure if I understood.
I think that we answer all the question, and we are running a little bit. I just want to do one curiosity about the low cranial irradiation.
Yep.
Can you say something about the side secondary effect?
Yeah.
Or something?
Yeah, absolutely. Yeah. So we lower the dose as much as possible. We know it does not lead to monocyte infiltration, so there's no opening of the blood-brain barrier.
Okay.
And we also analyze the double-strand breaks in microglia and in other cells. So we find a little bit of double-strand breaks of microglia, but it's like 1 order of magnitude less than in the rest of the hippocampus, because they are mitotically quiescent, so they are not really. So yes, they are affected, and we can see changes in the transcriptomic profile due to the irradiation unrelated to phagocytosis. So definitely it's not completely clean. Yes,
Okay,
absolutely right.
Thank you so much. So I just have to say that there are a comment here. Hi, Amanda. Javier Sánchez Perona from Instituto de la Grasa CSIC works with lipoproteins and microglia, just in case you want to.
Okay, great. Just send me an email. Thank you. Great. I have no expertise, so that would be great.
So yeah, I think we need a new seminar just for questions.
Yeah, well, thank you very much. It was a great feedback. Thank you very much.
It was great to have you here today. I think that we learned a lot. We have a lot of questions, and also about lipidomic. I know that Patricia Aspichueta also can help about that.
Yeah.
So thank you so much, Patricia.
Yes, thank you. Yes, thank you very much, Amanda and Patricia González, for this wonderful talk, and of course to all the people that were able to join us today, which has been a lot of people, a bunch of people. Amanda, you really
thank you.
A science star.
No.
So
thank you.
So thank you. It was fun to listen to you again, and well,
it was great seeing you guys. Thank you very much. Bye.
Thank you so much, Amanda. Bye bye.
Bye. Thank you.